Hi, I'm Norah Hughes, and I chose Jennifer Doudna as my role model. She grew up in a small town in Hawaii. She noticed a lot of biodiversity in the plants and animals around her. She then was curious about how they evolved to be that way, which she later connected to chemistry. She did grow up with a little bit of bullying and name calling, which she thinks helped her in her inner strength. She doesn't think that it was a good thing necessarily, but it did help her with future confrontations and insecurity. She completed a bachelor's in chemistry at the Pomona College in California in 1985 and completed a PhD in biochemistry from Harvard in 1989. Some of her hobbies include gardening, specifically tropical plants from Hawaii, like the strawberry guava to the right. She enjoys doing puzzles and reading, like all sorts of things, scientific and non-scientific. What I admire most about her is that she drives other women to stop self-doubting themselves. She thinks that a lot of people limit themselves, and especially women. She just thinks that you are capable of so much more than you think you are, which I just found very inspiring. As well, she's a female, and as a female, I love learning about other females in the field since predominantly in my education I've found that I've learned a lot about men. As well, we share a lot of the similar hobbies, including gardening, puzzles, and reading. And I am interested in genetics. I might pursue it in the future. Otherwise, I pursue plants. So I have a couple commonalities with her. So here's a quote that I took from an interview. I would never want to stereotype, but I do think there's more of a tendency by women and girls to underestimate themselves. We have to actively encourage both ourselves and other women and girls that might be following in our footsteps to actively put that little voice aside and trust that actually they're probably better than they think they are. It just really stood out to me and I loved it. So she works in biochemistry, specifically genes. She works on the CRISPR immune system, which is found in prokaryotes. She discovered the Cas9 enzyme, which is what cuts the RNA and DNA, which makes them ineffective. This would probably be what she's most known for. This can be used in gene modification by cutting the DNA and inserting or just turning off the gene. A recent publication with others, she studied live mice with a humanized immune system. So she used viral envelopes and put Cas9 enzymes and the gene of choice in there so they'd be delivery vehicles to specified T cells. So to the right, we have a figure. The first figure shows just the viral envelope and the gene and the enzyme, and it shows that they were trying to knock out the protein B2M, and the targeted cells, which is green, and the bystanders, which is blue, were both modified, and they were trying to target very specific ones. So they then added an antibody, which would target them, and this ended up majorly only targeting the specific cells and limiting the bystanders that were modified. And then lastly, on the bottom, we have the control with just the antibody, and there was no modification going on. And the overall figure is shown in the bottom right. So you can see that modification was happening for both the bystanders and targeted without the antibody. But when they added the antibody, only the targeted cells were being modified. Well, the bystanders a little bit, but it was greatly decreased. So the next steps would be to make vehicles more efficient by limiting the viral structure. They realized that the capsid might not be very necessary, and they want to apply this to non-immune cells. But overall, they succeeded in targeting specific cells and limiting other cells being modified. Thank you so much for listening to this presentation. I find Jennifer Doudna to be inspirational, and I think she will uncover loads more, even though she's accomplished so much already. Here are my information citations. Here are my photo hyperlinks. And the numbers correspond to the pictures on the slides. And lastly, here's a credit to the template I use from SlidesGo because it's very pretty.